gram-negative bacilli. So there's actually a lot of them. Ayan. So, ako rin po yung gumawa ng voice-guided presentation for Chapter 18 na naka-upload po sa Canvas nyo. So, doon po, na-discuss ko yung 3 most clinically significant na Haemophilus species, plus yung mga miscellaneous, plus yung HASIC group. And then later, or at the later parts of the discussion, we talked about the fastidious gram-negative bacilli. So all in all, that was worth 105 slides. Okay? So dito po ang i-discuss na lang natin is Haemophilus influenzae, aegyptius, ducreyi, and then some of the miscellaneous species. Tapos, skip over na po natin yung HASIC. Proceed na tayo agad dun sa mga clinically significant fastidious gram-negative bacilli. Alright, so ayan po. For the general characteristics of Haemophilus species, they are gram-negative and pleomorphic, meaning that even though we have established the fact that they are bacilli in form, they could still vary in appearance. Diba po? May mga previously discussed organisms naman na rin tayo na classified as pleomorphic. So hindi po pare-parehas or hindi consistent yung itsura nila upon microscopic examination. So in direct smears from clinical samples, they could be seen as short cocobacilli, just like these ones in this photo. So ayan po. And then, if ang gagawin naman natin is we first take the clinical specimens and then make cultures out of those, and then parang microscopically observe those organisms na nanggaling na sa culture, mag-iiba rin po yung itsura nila. So they will appear like long filaments as shown here. So dito po, kung direct from clinical sample natin sila observe, makikita natin na parang magkakaiwalay sila. But if we were to culture them first before making stained smears, makikita natin na mas joined together yung mga cells, forming filaments. Now, Haemophilus species are also non-motile, facultative anaerobes, and some of them are part of the normal flora of mucous membranes in humans. So mamaya po pag inisa-isa natin yung bawat species, malalaman natin kung sino yung pwedeng maging part ng normal flora and sino yung hindi talaga part ng normal flora. So pag na-observe natin itong mga to, most likely they are the causative agent of the disease. Alright, so ayan po. There are 8 species that have been associated or that have been isolated from humans. Okay, so there's a total of 8, whether they are pathogenic or commensal. And then, ayan, take note that Haemophilus influenzae and Haemophilus aegyptius have their own common names. Okay, so Haemophilus influenzae is also known as the Piper's bacillus, and Haemophilus aegyptius is commonly known as the Cockweek's bacillus. And so out of those 8 species, we have 3 that have been recognized clinically significant, and these are Haemophilus influenzae, which could cause meningitis and epiglottitis. And then there's also, nag-glitch yung camera ko. And then there's also Haemophilus aegyptius, which is a causative agent of pink eye. And last one is Haemophilus ducreyi, which could cause sexually transmitted infections such as chancroid or soft chancre or the GUD. So dali lang po. Alright, so ayan, tuloy po tayo sa general characteristics. Some of the above-mentioned Haemophilus species are normal flora of the upper respiratory tract of healthy people. So non-encapsulated strains of Haemophilus influenzae have been isolated in children, while for adults, about 10% of our normal flora are composed of Haemophilus parainfluenzae, as well as other non-encapsulated Haemophilus influenzae. All right, so now that we know or how to classify Haemophilus, or since we now know that Haemophilus species are classified as fastidious organisms, ano-ano po yung mga growth factors na nire-require nila. Alright, so their genus name, Haemophilus, was derived from the Greek words haima, meaning blood, and philia, or loving, which would directly translates to blood lover, which makes perfect sense considering that they require nutrients that are present in the blood for them to grow. So there are two growth factors present in the blood. First is the X factor, or the unknown factor, which we now know as hemin or hematin. So hindi naman na talaga siya unknown. And then we also have the V factor, or the vitamin factor. Specifically, this is composed of nicotinamide adenine dinucleotide, or NAD for short. Okay, so the V factor can only be found within RBCs. Hence, ang V factor po ay hindi magiging accessible sa isang organism if the RBCs on that culture media are still intact. Okay? Now, there are species that do not need both of these growth factors. So may mga species po na ang kailangan lang nila is X factor lang. Yung iba naman po ang kailangan nila is V factor lang. And those that only need the V factor have been conveniently named as with those with the prefix para. So ayan po, Haemophilus para influenzae, Haemophilus para hemolyticus, Haemophilus para afrohemolyticus. So ayan po, as shown here on the lower left photo, their growth was observed only next to the strip lines, the V factor. So mamaya po i-discuss din natin paano ginagawa itong method of testing na to. Alright, so both the X and V factors are found within red blood cells, although only the X factor is readily available. So kahit hindi i-hemolyze yung, or kahit gumamit tayo ng culture media kung saan hindi hemolyze yung mga RBCs, X factor lang po yung magiging available to the cells. Now, for some Haemophilus species, some of them are able to acquire the V factor, and the RBCs in the culture media needs to be hemolyzed for them. Now, this means that they cannot be grown on BAP or SBA. Rather, chocolate agar is needed to isolate these organisms. So, mamaya pa makikita natin na hindi lang sheep blood yung pwedeng gamitin for blood agar plates. As long as hemolyzed yung red blood cells, we could make blood agar plates. Now, in the laboratory, na-discover po nila yung phenomenon of satellitism. So for organisms that need both the X and V factor, ma-observe pa rin naman po natin yung growth nila on blood agar plates. Paano? So we may be able to observe what is known as satellitism. So as previously mentioned, X factor requiring Haemophilus cannot grow on SBA, at least not by themselves. So for example, Haemophilus influenzae is straight right next to a beta hemolytic organism such as Staphylococcus aureus. Okay, so the organism would then cause lysis of the RBCs in the medium. So yung Staph aureus, yung nag-hemolyze ng RBCs dun sa medium, which would now release the V factor within the RBCs, making them available to the Haemophilus, and thus growth would be observed in the immediate surrounding of the Staphylococcus strain. So again, this phenomenon is known as satellitism. So in this given example, ito po, by itself, hindi kaya ng Haemophilus influenzae na mag-grow sa SBA or BAP lang. Pero since katabi niya yung Staph aureus na nagpo-produce ng, or yung Staph aureus that can hemolyze the RBCs and produce NAD, kaya na po niyang mag-grow sa immediate surrounding lang ng Staph aureus. Kasi doon lang naman yung parts ng agar kung saan available na yung V factor. So this is what we meant when we say satellitism. Okay, let's now proceed with the specific species. First up is Haemophilus influenzae, commonly known as the Piper's bacillus. Okay, so Haemophilus influenzae's most significant virulence factor is its capsule. And this would remind us of Streptococcus pneumoniae, di ba po? The causative agent of lobar pneumonia. parehas silang merong polysaccharide capsule. So the polysaccharide capsules that make up this one has served as the basis for serologic grouping. So yung mga polysaccharides that make up the capsule, yun po yung tinitignan to serologically group the different types of Haemophilus influenzae. And the most invasive infections were attributed to Haemophilus influenzae serotype B. So in unvaccinated children, this has been a causative agent of meningitis. Now, not all strains of Haemophilus influenzae are encapsulated, and without their capsule, they cannot be serotype. Okay, so wala silang serologic group, and they would simply be referred to as the non-typable Haemophilus influenzae, or the NTHI. So, for short, yung Haemophilus influenzae serotype B po, this is otherwise known as the HIB or HIB. Hindi V, or it's B. Okay, so HIB. All right, so aside from the capsule, Haemophilus influenzae has a few other virulence factors. First up is immunoglobulin A protease, which could destroy secretory IgA. IgA is usually found in the secretions of mucous membranes, which would provide protection from pathogens along those areas such as the respiratory epithelium. So it is because of these IgA proteases that Haemophilus influenzae can successfully colonize the host upper respiratory tract. Before we proceed, recall time. Recall time po tayo. If you know, as usual po, if you know the answer, you may type sa chat box natin. So recall time, which gram-negative, obligate aerobe capnophilic organism also produces IgA protease as one of its virulence factors? Sige po, if anybody remembers. Which gram-negative, obligate aerobe capnophilic organism also produces IgA protease as one of its virulence factors? One of the clues this is a causative agent of either sexually or vertically transmitted disease. And this is a gram-negative diplococci. Okay, so the correct answer is Neisseria gonorrhea, as shown here in this pop-up screenshot. Ayan. So, kapag sexually transmitted po, it would cause gonorrhea. Pero kapag vertical transmission, it would cause ophthalmia neonatorum. Okay, so parehas po sila na nagpo-produce ng IgA protease. Okay, proceeding to the next virulence factor. And another virulence factor are their adherence mechanism. So this is specifically for the non-typable Haemophilus influenzae. Okay, so since NTHIs are adherent to whichever surface they are colonizing, they tend to cause more localized infections. Example, acute conjunctivitis, as compared to encapsulated Haemophilus influenzae that are non-adherent and would most likely cause systemic infections. Okay? Yung capsule po kasi ng organism, nagpe-prevent siya na maging adherent yung bacterial cells. Instead, ang nangyayari po is it could facilitate disseminated infections kasi hindi nila kaya mag-adhere to one place. So for the non-typable Haemophilus influenzae or yung mga NTHI na walang capsule, kaya nilang mag-adhere to surfaces. So i-expect natin na localize yung mga infections na kaya nilang i-cause. And lastly, Haemophilus influenzae also has outer membrane proteins that paralyze the sweeping motion of the ciliated respiratory epithelium. All right, so here is the list of infections that may be caused by both encapsulated and non-encapsulated Haemophilus influenzae. So notice that the encapsulated strains cause generally systemic diseases such as septicemia or arthritis, while the non-encapsulated strains with adherence mechanisms will cause localized infections like conjunctivitis or otitis media. So naka-confine lang po sa isang lugar yung infections na dinudulot nila. Hindi tulad ng mga to. Septicemia, it's in the patient's blood. Meningitis reaches the brain. Arthritis could happen anywhere on your joints. So, ayan po. All right, let's now talk about these clinical manifestations. So, meningitis is due to serotype B Haemophilus influenzae meningitis due to this organism used to be very rampant before vaccines were available against this. So here in the Philippines, we have what is known as the pentahib vaccine. Okay, so meron naman po tayong bakuna laban dito. The pentahib is a pentavalent vaccine, which means that this is effective against 5 different diseases contained in one shot. Okay, so this pentavalent vaccine gives protection against diphtheria caused by Corynebacterium diphtheriae. It could protect against tetanus caused by Clostridium tetani. It could protect against pertussis caused by Bordetella pertussis, polio caused by the polio virus, and other diseases or meningitis caused by Haemophilus influenza type B. So again, yun po yung contents ng pentahib vaccine. Now, for meningitis caused by Haemophilus influenzae, the symptoms would start off very non-specific. Okay, so there's only a mild respiratory disease with headache and stiff neck before the patient eventually begins to show signs of meningitis because, as mentioned earlier, encapsulated strains of Haemophilus influenzae do not have adherence mechanisms, and therefore it is not unusual for them to spread from the upper respiratory tract papunta po sa central nervous system. Ayan, next po is epiglottitis. So Haemophilus influenzae can also cause epiglottitis or the inflammation of the flap of cartilage at the base of the tongue. As we can see right here, dito po sa, ayan po, meron namang arrows na nakaturo dito sa mga given photos. All right? So epiglottitis has a rapid onset. So mabilis yung development of disease, and it can cause complete airway obstruction, especially for children since their airways are much smaller. Okay, so this photo on the right shows a significant size difference between the normal epiglottis, so ito po yung normal, and an inflamed epiglottis. So as you can see right there, it's thicker. So mas makapal na yung epiglottis ng pasyente. So parang instead of doing its job, doing its purpose, ibinablock na lang niya yung airway ng pasyente. Now, in relation to the next disease that we will be discussing, I would like you to take note of where the trachea is, which is right here. Ito po, yung passageway na to. Yan po yung trachea ng pasyente. Okay? So, both radiographs, we can see na normal naman po yung trachea ng pasyente. Ang problema lang po is swollen yung epiglottis ng pasyente. So, ayan po, normal trachea is unobstructed, and ayan po, i-compare natin siya dun sa next disease. So, bacterial tracheitis. In bacterial tracheitis, the patient accumulates thick secretions that can occlude the trachea, and it is just as fatal as epiglottitis. Okay, so parehas din po kasing nababarahan yung airways ng pasyente sa epiglottitis and bacterial tracheitis. Ang pinagkaiba lang nila is magkaiba yung area ng upper respiratory tract na affected. So as you can see right here, ayan po, mas makitid na yung passageway. Alright, so differentiating this with epiglottitis, in this case of bacterial tracheitis, the epiglottis is normal in size, as you can see right here. So ito po yung epiglottis ng pasyente. So normal in size, hindi siya as thick as the first one that we saw earlier. But the trachea almost does not have much space left for the air to pass through. So yun po yung difference between epiglottitis and bacterial tracheitis, which could both be caused by Haemophilus influenzae. All right, so now let's discuss the next medically significant Haemophilus species on our list, and that is Haemophilus aegyptius, also known as the cockweek's bacillus. So Haemophilus aegyptius was named for obvious reasons. Okay, so meron siyang historical background. It was first isolated from cases of conjunctivitis or exudates from the conjunctiva of Egyptian patients. Okay, so acute purulent conjunctivitis is more commonly known as pink eye because of how pinkish the small blood vessels in the conjunctiva appear when they are inflamed, as you can see here in the photos to the right. So ayun po, according to the Centers for Disease Control and Prevention, the symptoms of pink eye would include the pinkish or reddish discoloration of the sclera of the eyes, and then there's also increased tear production, and also there's a constant feeling of a foreign body that is stuck within the eyes, and there is obviously itching, irritation, and burning, and then there is discharge production consisting of pus and mucus, and there's also crusting of the eyelids or eyelashes, especially in the morning. So parang nagmumuta ng sobra yung pasyente. Ayan, next po is Haemophilus influenzae biogroup aegyptius. So this next pathogen is actually a different species from the Haemophilus aegyptius group, or Haemophilus aegyptius species. So this is the Haemophilus influenzae biogroup aegyptius. Okay, so this is the causative agent of Brazilian purpuric fever, and its symptoms include recurrent conjunctivitis plus high fever and rashes, and this could progress to septicemia and vascular collapse. So ayan po, medyo mataas yung mortality rate in untreated patients. Mortality rate could reach up to 70% within 48 hours of onset. And the next clinically significant Haemophilus on our discussion is Haemophilus ducreyi. So this is not a part of the normal flora, and this could be only isolated from humans during the course of infection. Ayan. So Haemophilus ducreyi is the causative agent of chancroid or soft chancre. So please do not confuse this with hard chancre, which could be observed in cases of syphilis. Okay, so mga next week pa po natin madi-discuss yun. Syphilis is caused by Treponema pallidum. Okay, so again, Haemophilus ducreyi causes soft chancre or chancroid. Incubation period would range from 4 to 14 days upon initial exposure. Now, the disease that it causes is referred to as the genital ulcer disease, or GUD. Now, because the chancroid facilitates the transmission of other STDs, all patients who have GUD should also be tested for human immunodeficiency virus and also syphilis and also the herpes virus. Kasi po yung mga ulcerations that are being produced in the infected areas, this could then serve as a passage for all these sexually transmitted infections to enter the host. Okay, so parang nagiging daan po nila yung mga ulcerations that has been produced during the course of infection nitong Haemophilus ducreyi. Right, so Haemophilus ducreyi is not part of the normal flora. So after an incubation period of approximately 4 hanggang 14 days, a non-indurated or parang nakalubog na painful lesions with an irregular edge would develop generally on the genitalia or perianal areas. And the most common sites of infection are on the penis and on the labia of the vagina, as you can see here in these examples. So pus-forming, enlarged, and draining inguinal lymph nodes would also form, and they are referred to as buboes. Okay, so they're commonly seen in most infected patients. So mamaya po, pagdating sa sample collection, pwede rin tayong kumuha ng specimen galing dun sa mga inflamed lymph nodes na yun. And let's now look at two other miscellaneous Haemophilus species. So Haemophilus para-influenzae, nabanggit na po natin to kanina, di ba? Along with, along with other parts of our lecture, what did we say about the Haemophilus species with the prefix para? Di ba po? Those with the prefix para, they only require the V factor. So Haemophilus para-influenzae can be isolated from the oral cavity of humans. So ito po, pwede siyang ma-isolate as normal flora of the oral cavity, and if given the opportunity, they may be able to cause endocarditis, specifically along the mitral valve or of the heart. So meron din siyang specific area na ini-infect, which is the mitral valve. So from the oral cavity, 1 month after an improperly performed dental procedure, and they can migrate to the heart and cause pathogenicity. So yun po yung nagiging way kung paano sila nanggagaling sa oral cavity at napupunta sa heart ng pasyente. Next is Haemophilus parahemolyticus. So all we have to know here is that this can commonly be found in the upper respiratory tract of humans, and it may sometimes cause pharyngitis. So itong dalawang to, hindi po siya ganun ka-clinically significant. Kaya it's only known as miscellaneous species. Kasi nga po, part sila ng normal flora, and it's not always that they would cause an infection. All right, so now, what are the methods of laboratory diagnosis for the different Haemophilus species? Ayan. So, unahin natin yung mismong method of specimen processing and isolation, which includes collection. Okay? So, here is a list of possible sources of specimen for any of the previously discussed infections caused by the different Haemophilus species. Now, if the physician suspects that it is a case of meningitis, then the appropriate specimen would be CSF. Bronchial washings would be recommended if the disease is respiratory in nature. So, ayun po. Ganun din naman po dito, no? Kung nasaan nagmamanifest yung disease, dun manggagaling yung specimen. So, for sexually transmitted infections, vaginal swabs, or abscess discharge, yung kukunin. Right? So it is important to remember that Haemophilus species are fastidious, meaning they require additional growth factors for us to successfully recover them from clinical specimens. So most routine culture media do not support the growth of Haemophilus species. Hence, they will not grow on McConkey agar. Okay? And nabanggit na rin natin kanina, most likely hindi sila mag-grow on BAP. So they will also die rapidly if not transported properly or processed immediately. So katulad din ng Neisseria species, they are very susceptible to drying and other harsh environmental conditions. Ayan po. Now, specimen collection for Haemophilus ducreyi. In collecting specimens for suspected cases of Haemophilus ducreyi, everything needs to be sterilized because our goal is to isolate the organism. Therefore, there should be as little to no contamination of organisms if possible. So, iiwasan natin na together with the sample is makakuha tayo ng normal flora or other contaminants from the environment. Now, the most important thing that you have to keep in mind here is that if collecting swabs for the isolation of Haemophilus ducreyi, we have to swab at the base of the ulcer or yung pinakailalim po nung abscess, okay? Because at the base of the ulcer is where most of the organisms will be found. All right, and then the preferred culture media would be chocolate agar or the Nairobi biplate medium. So pwede naman na po yung standard na chocolate agar. But if you need additional supplementation or a special type of culture media, ayan po. The Nairobi biplate medium has been formulated specifically for the isolation of Haemophilus ducreyi. And since fastidious nga po sila, medyo slow growing din po sila. So duration of incubation would range up to 7 days. Ayan, so ito po. Listed here are the contents of the Nairobi biplate medium. So the purpose of vancomycin is to reduce the growth of commensal bacteria from the patient's skin. Kasi po, as much as we always try to eliminate the possibility of um, acquiring normal flora upon specimen collection, minsan po hindi may iwasan, may sasama at may sasama pa rin na normal flora. So yun po yung purpose ng vancomycin dito. Okay, so again, vancomycin is to reduce the growth of commensal bacteria from the patient's skin. And then, while some normal flora are inhibited, Haemophilus ducreyi will continue to grow since ang Haemophilus ducreyi po is resistant to vancomycin. And then, ayan po, nakalista din po dito yung iba pang sources of nutrients. And let's now talk about its colony morphology. So the colony morphology of Haemophilus ducreyi appears to be small, flat, non-mucoid, tan to yellow colored colonies. And if you take a clean inoculating loop and try to push one of the isolated colonies from the side, they will move but will remain intact, which would remind us of Moraxella catarrhalis. Di ba may ganun din pong characteristic yung Moraxella catarrhalis, which was able to earn them the nickname of being like hockey puck colonies. Kasi kapag kukuha ka ng colonies, imbis na makuha mo nga mismo yung colonies, they would just move from one place to another. So minsan po, ma-observe din natin yan with Haemophilus ducreyi. Ayan. Next naman po, for Haemophilus influenzae, the recommended culture media is the horse blood bacitracin agar. So although for most laboratories that don't have that chocolate agar infused with bacitracin may also be used. Okay? So they must be incubated under capnophilic conditions. So ayan, naka-highlight naman na po dito lahat ng kailangan nating tandaan. For Haemophilus influenzae, they are not necessarily slow growers. Okay, so fastidious sila, pero hindi sila slow growing. Duration of incubation would only be for up to 18 to 24 hours. So after 24 hours, expect na natin na makakapag-view po tayo ng colonies kung Haemophilus influenzae po yung isolate. Ayan, and nabanggit din po dito, for Haemophilus aegyptius, the preferred culture media is an enriched chocolate agar that has been supplemented with iso-Vitalex. So this is something na nabanggit na po natin during our discussion with, again, Neisseria, di ba? Ang dami nilang common factors. So for Haemophilus aegyptius, pwede nyo rin pong isupplement yung culture media nila with iso-Vitalex. Medyo mas slow growing or medyo mas matagal po yung incubation period for Haemophilus aegyptius. Duration would take about 4 days bago po tayo makapag-observe ng colonies. Alright, colony morphology of Haemophilus influenzae. The appearance of Haemophilus influenzae on chocolate agar is described as non-hemolytic, tan, convex with a mousy or bleach-like odor. So isa po yun sa mga characteristics ng Haemophilus influenzae. So if they produce a bleach-like odor or it smells like a place that is infested with mouse or mice, yun po, yun yung sisabing mousy odor. Ayan po. Dapat maisip na natin agad na most likely Haemophilus influenzae po yung isolate. Alright, and then its biogroup aegyptius may also appear similarly. So di ba po, yung nabanggit natin kanina, yung Haemophilus influenzae biogroup aegyptius, ganun din po, ganito rin yung itsura niya. Alright, colony morphology naman ng Haemophilus influenzae pa rin, if the strain has been the strain that has been isolated is encapsulated, expect that the colonies will appear larger and much more mucoid compared to the NTHI strains. Okay, so isoli ko lang yung previous slide. And po, kapag NTHI, most likely, ganito po yung itsura. Pero pag encapsulated yung strains, expect natin na mucoid and larger yung itsura ng colonies. So an example of this has been shown right here on these photos. So ayan po, these are examples of encapsulated strains of Haemophilus influenzae. Ayan, nabanggit din po dito, for Haemophilus parainfluenzae, colonies will appear dry and slightly larger in comparison to Haemophilus influenzae colonies. And then kapag parahemolyticus naman po, Haemophilus parahemolyticus may resemble parainfluenzae, but do take note that parahemolyticus is beta hemolytic, as the name implies, no? Parahemolyticus. So it could cause beta hemolysis. Alright, tignan naman po natin yung microscopic morphology nila. Looking at the microscopic morphology of encapsulated Haemophilus influenzae, we may be able to observe the appearance of halos, which are non-staining areas that surround each cell seen in this photo to the right. So ayan po, di ba? Merong clear na area sa paligid ng bawat isang cell, and that is because they are encapsulated. Ayan, ito naman po, for Haemophilus ducreyi. So when it comes to the microscopic morphology of Haemophilus ducreyi, there are two characteristic descriptions that may be observed. First are the railroad tracks, okay? In Tagalog, riles ng tren. So like this one, they are arranged linearly. Ayan po. So yun yung tinatawag na railroad tracks. Parang nakapila sila. Okay, and then next one would be school of fish. Okay, so they are grouped together or just clustered together in no particular order, just like this one. So ito po yung tinatawag na school of fish. And let's now talk about the X and V factor requirement. So we would like to check their X and V factor requirement. Okay, so example, nakapag-isolate na nga po tayo or nakapag-observe na tayo ng colonies after plating them. Now, we would like to know kung sila ba ay X factor dependent, V factor dependent, or both. So the usual approach to this is either by is by placing strips on an agar plate. So ganito po yung itsura ng mga unang slides na pinakita ko kanina, di ba? If growth is observed around only the X strip, it means that the organism only requires the X factor, as you can see right here. If the organism grows around only the side of the V strip, then it requires only nicotinamide adenine dinucleotide, as we can see right here. Pero po, if our isolate grows right in the middle of both the X and V strip, ayan, this means that the Haemophilus species would require both the X and V factor. So madali lang naman po tandaan. Simple lang naman po i-interpret yung X and V factor requirement. Kung saan sila nag-grow, yun yung requirement or yun yung factor na nire-require nila. Kung nag-grow sila right in the middle, it means dun kasi present both factors. So yun kailangan nila both factors. Alright? Now, and po, aside from the paper strips, we may also use discs. So ayan po. In this example, a separate disc is already available for both the X and V factor. So hindi na natin kailangan tignan yung middle portion ng bawat disc tulad itong X factor, V factor. Dito po, meron ng hiwalay na disc na kinocontain niya both yung X and V factor. So kung dun nag-grow yung bacteria, then most likely, kailangan niya both factors. And po, this is what it looks like in person or in real life. And in more advanced and highly funded laboratories, they have what is known as the Haemophilus quad plate. So as the name implies, the quad plate is sliced into four, or meron siyang four different sections. All right. Now, there are four different zones to streak our bacterial isolate on. So there is an agar base infused only with the X factor. Meron naman pong agar base that is infused only with the V factor, and there is a quadrant kung saan both X and V factor nandun. Plus, there is also a quadrant that contains Ayan. Horse RBCs. Okay, so on this quadrant, the organism's ability to produce hemolysis is observed. So, example, Haemophilus parahemolyticus po yung isolate nyo. So, if nag-produce siya ng growth and hemolysis dito sa area na to, then dapat po maisip nyo na most likely Haemophilus parahemolyticus yung isolate nyo. So, it's like a 4-in-1. You can observe 4 different things in one plate. So, ayan po, yung Haemophilus quad plate. All right, so ayan po, on this table, we see both the clinically significant and miscellaneous Haemophilus species alongside the growth factors that they require. So naka-table format na po if ever yan yung preferred learning method nyo kung mas madali nyo pong natatandaan with tables. So Haemophilus influenzae, Aegyptius, and Influenzae biogroup Aegyptius would both require both the X and V factors, while Haemophilus ducreyi requires only the X factor. All right, so again, ang X factor po, this contains hemin or hematin. And then ang V factor po, this contains nicotinamide adenine dinucleotide or NAD. Ayan, next na po is the porphyrin test. So another test that can be done for us to identify Haemophilus species is the porphyrin test. So the principle of this test is that if the organism can convert delta amino levulinic acid into porphyrins or porphobilinogen, then this means that they are not X factor dependent. Okay, so ulitin natin yon. If the organism can convert delta amino levulinic acid into porphyrins, this means hindi nila kailangan yung X factor. Okay? So these substances are detected by adding paradimethylaminobenzaldehyde, commonly known as the COVAX reagent. And then we observe this under ultraviolet light with the use of a Woods lamp that is set at a wavelength of 360 nanometers. So tignan po natin yung itsura niya on the next slide. Ayan. As shown in this picture, the organism on the top is porphyrin negative, and the streak at the bottom is positive for porphyrin or either porphyrin or porphobilinogen production. We can't really differentiate kung porphyrin or porphobilinogen ba yung produce niya, but if it was able to produce this type of fluorescence, at least we know na positive siya sa porphyrin test and hindi niya po kailangan yung X factor. All right, so let's explain that further. Species that are porphyrin negative cannot synthesize heme. Hence, they would rely on being provided with the X factor for them to grow. Now, species that can synthesize these intermediates, for the X factor does not need it anymore and will only require supplementation of the V factor. Kasi po, parang in simpler terms, kaya nilang gumawa ng sarili nila. Okay, so they can synthesize their own intermediates for the X factor. So hindi nila kailangang umasa dun sa culture media to provide them with the X factor. So yun po yung principle ng porphyrin test. All right, so in summary, again, if the organism is X factor dependent, it will be porphyrin test negative kasi kailangan pa niya ng porphyrin. But if the bacterial isolate requires only the V factor, then it will be porphyrin positive kasi kaya niyang mag-produce ng sarili niyang components for the X factor. All right. Sige, let's have 10 minutes break. Let's have 10 minutes break. Tapos po, let's start with all the other species na hindi Haemophilus at hindi members ng HASEC. Okay? So, saktong-sakto, it's 9 o'clock. Let's all come back here at 9:10. Okay? Nga pala guys, yung mga hindi pa nakakapag-view ng grades nila, if you would like to see your grades, pero hindi kayo nakapag-attend nung, or hindi kayo nakapag-message nung scheduled consultation, mag-message na lang po kayo, mag-direct message na lang kayo dito after nung discussion para po masend ko ulit or masend ko pa sa inyo yung grades ninyo. Okay? For those who haven't seen theirs. Thank you. Alright, so ayan, resume na po tayo with the discussion. Tapos na tayo with Haemophilus species and other species na um, gram-negative fastidious pero hindi belong sa Hasik group. Alright, so let's start off with Capnocytophaga. Alright, so there are 5 species that can be found as a normal flora of the human cavity as listed here. Okay, so they require a capnophilic setup, as the name implies, diba? Capnocytophaga. So we would expect that they are capnophilic. And then, ayan, they can cause septicemia in patients with low neutrophil counts, which is termed as a neutropenia. So, kapag neutropenic po yung pasyente, ayan po, expect natin na mataas yung chance that they would be able to, or they would most likely develop an infection that is caused by capnocytophaga. Alright, as for their colony morphology, although Capnocytophaga is non-motile, okay, keep that in mind, Capnocytophaga is non-motile, their colonies would display a gliding motility. So parang merong motility pattern yung colonies nila upon culturing. Okay, so most of them are non-hemolytic, except for one species that has been observed to be beta hemolytic and has been aptly named Capnocytophaga hemolytica. So in microscopy, they resemble Fusobacterium due to their pointed ends. Alright, so ayan po, ito yung tinatawag na gliding motility ng Capnocytophaga species. Next is Pasturella. Ayan. They're found normally on the upper respiratory tract of both mammals and birds. Okay, so these are fastidious gram-negative cocobacilli that tends to resemble safety pin due to their bipolar staining. All right, as you can see here on this bottom left photo, ayan, the ends of each cells are stained more intensely compared to the rest of the cell. So that has something to do with their cell structure. Kaya po mas nagko-concentrate yung stain sa bawat ends nung cell. Kaya po siya nabigyan ng parang safety pin appearance or bipolar staining. Ayan po. Now, the most common isolate for this genus is Pasturella multocida. Hindi na po natin iisa-isa ito kasi hindi naman din sila ganun ka clinically significant. And so pasteurellosis is a zoonotic disease, meaning this could be acquired from infected animals or animal products. So yun po yung meaning ng zoonotic diseases, di ba po? If we acquire them in a way that, or in any method that involves contact with animals or infected animal products, then this is a zoonotic disease. And the most common form is cutaneous pasteurellosis. Now, at times, these may also lead to systemic infections. And over here are the typical biochemical test results. Ayan po. So actually, there's a lot. Hindi madaling tandaan yung mga biochemical test results nila, lalo na po pag ganito kadami, di ba? So keep in mind its other characteristics. So kapag nabanggit dun sa case na is isang zoonotic disease, and nabanggit po na they appear to have a bipolar staining pattern or they appear to look like safety pins, ayun po, dapat maisip nyo na agad, is the Pasturella species, just in case nahirapan po kayong tandaan lahat ng biochemical characteristics niya. Right, colony morphology of Pasturella on sheep blood agar appears mucoid after 24 hours of incubation. But if we were to extend the incubation to 48 hours, the colonies would now produce a green to brown colored halo. So as you can see right here, ayan, medyo mas mukha siyang brown dito sa picture na to. So kumbaga, as time passes, nagbabago po yung itsura ng colonies ng Pasturella species. Next is Brucella, otherwise known as the Banks bacillus. Ayan. Take note that the Brucella species is listed as a Category B biological agent by the Centers for Disease Control and Prevention. So Brucella may be used as a bioterrorism weapon. Recall time, hindi ko to pinlano, pero recall time po, Ano yung isa pang organism na discuss natin last time which has been classified as a category A bioterrorism agent? Nakalimutan na. It's Bacillus anthracis, okay? Ang Bacillus anthracis po, which could cause either cutaneous or pulmonary or other forms of anthrax, ayan po. This has been classified as a category A bioterrorism agent. There are three categories of bioterrorism agents. So yung A po, yung pinakamataas or the highest importance, B, and then C. Yung C, yung least important. All right. Now, brucellosis is also a zoonotic disease katulad ng pasteurellosis. So dito po sa discussion natin, marami tayong madadaanan ng mga zoonotic bacterial infections. So isa na yung Pasteurella species. Now, ito po, Brucella species. This is also a zoonotic disease that can be found worldwide and is known as the cause of the undulant fever or the Malta fever. So bakit siya tinawag na undulant? Unahin muna po natin yung modes of transmission. Okay? So modes of transmission include either aerosols, inhalation of aerosols, or oral or percutaneous routes. Okay, and there are three clinical stages of brucellosis. So first is the acute stage, which happens within 1 to 4 weeks after exposure. So parang yun po yung pinakaunang incubation period niya. Okay? 1 to 4 weeks after exposure, magsisimula po yung acute stage. So the symptoms here are non-specific, which includes fever, headache, anorexia, and myalgia. So as you already know, there are 100 different other causative agents of those symptoms. Next is the subchronic or undulant form, where, or this is the characteristic manifestation of brucellosis. Okay, the patient now experiences undulant fevers where their temperature is normal during the day, but it rises during the afternoon or the evening. So pabago-bago or hindi steady yung pag-angat ng temperature ng pasyente. Sa umaga normal, sa hapon or gabi, medyo mas mataas or parang sa evening nilalagnat yung pasyente. And then the chronic stage of brucellosis happens a year after exposure. So brucellosis could be considered a chronic disease kasi po kaya niyang mag-cause ng pathogenicity that could last up to a year. Okay, so systemic symptoms such as arthritis may be observed. All right, so there are 4 species that have been associated with human infections, and these are Brucella melitensis, abortus, suis, and canis. Now, according to our reference textbook, there are 2 additional species that have been isolated from marine mammals. But for the most part, itong 4 lang po yung pinakakailangan natin tandaan. All right. Now, for the general characteristics, this is the same as the previously discussed organisms. No, kaya po hindi natin inuna yung general characteristics ng Brucella species kasi parang hindi naman siya ganun ka parang hindi siya ganun ka-unique or hindi naman ganun ka distinguishing yung general characteristics ng Brucella as compared to all the other genus or genera that we have discussed earlier. So they're also non-encapsulated, they're also non-spore forming, and they must be handled at a biosafety level 3 setup because, again, this organism could potentially be a bioterrorism agent, specifically a Class B bioterrorism agent. Now, acceptable clinical samples for the isolation of Brucella include blood and bone marrow. Ayan po. Now, the appropriate culture media would include sheep blood agar, pwede rin pong chocolate agar, as well as the modified Thyer Martin and the Martin Lewis agars, which, if you would remember, originally ginagamit po sila for Neisseria gonorrhoeae. Okay? Now, these agars are typically used for the isolation of Neisseria. Now, colonies may be observed in as early as 18 hours of incubation, but before reporting as negative plates should be kept for as long as 4 days. So aside from being fastidious, minsan po yung isolates natin pwede rin maging slow growers. So within 24 hours, if wala pa po tayong na-observe na colonies, pero suspect natin na Brucella species yung isolate, we should extend the incubation period for up to 4 days. And po, now straight off from our textbook, and po, here is just a table showing us the differential characteristics of Brucella species that can cause human infections. Guys, do take note that hindi po porket example, ako yung gumawa nung voice-guided presentation sa Canvas module natin. It does not necessarily mean na ako po yung gumawa ng quizzes nyo. Okay? So, randomly distributed po kasi yung assignment ng mga contributions sa amin. So, although ako yung gumawa ng discussion for Chapter 18, pwedeng ibang professor po yung gumawa ng questions for Chapter 18. So, ganun po kami dito sa Micro 1. Sa Micro 2 kasi, ako yung course coordinator nila doon. So, ang ginagawa ko doon, kung sino yung naggawa ng lecture, siya rin yung magagawa ng exam. Dito po, hindi. So, maaari po na may mga hindi ako na-discuss doon sa voice-guided presentation or dito po sa discussion natin ngayon, pero itatanong doon sa quiz kasi hindi ako yung gumawa ng quiz. So, I recommend na i-check nyo pa rin po yung mga question or yung mga tables na ganito po sa book ninyo. Just in case may lumabas po ng mga questions galing sa book na hindi po natin nadaanan dito sa discussion. Alright? Ayan, next po is Francisella tularensis. So Francisella tularensis has 3 subspecies, namely Francisella tularensis subspecies tularensis, and then subspecies holarctica, and then subspecies mediasiatica. Alright, so tulad ng Brucella and tulad ng Pastorella, ayan po, Francisella is also a zoonotic pathogen that causes tularemia, also known as the rabbit fever. Okay, so the most common form of tularemia is ulceroglandular. So, tulad ng ibang mga iba kasi silang forms, pero ayun po, tandaan po yung mga common forms nila. So, for tularemia, its most common form is ulceroglandular. And an ulceration would form at the inoculation site, and the lymph nodes within its proximity would become enlarged. So may makakapag-observe po tayo ng ulceration. Example, kung saan na-inoculate yung organism. Okay, so there are many other forms of tularemia such as pulmonary, oropharyngeal, oculoglandular, and so on. So just as previously tackled, or such as the previously tackled organisms, Francisella tularensis is also considered as a potential bioterrorism agent placed at a category A by the CDC. Okay, so kung yung brucellosis po, this is considered as a category B agent. Ang Francisella po, medyo mas mataas yung classification na. It's a category A bioterrorism agent. All right, for the general characteristics of Francisella tularensis, this is a small, non-motile, non-spore-forming strict aerobe. And as a fastidious bacteria, it requires the following sources of nutrition. So cysteine or cystine and thiosulfate. So itong tatlo po yung pinaka-requirement niya as a supplementation for us to be able to isolate this organism. And listed here are the appropriate culture media for the isolation of Francisella tularensis. And note that this organism will also not grow on McConkey and cannot be grown on McConkey and will also not grow on the Eocene methylene blue agar. So ayan po, MAC and EMB agars do not support Francisella tularensis growth. Tandaan po na ang MAC and EMB, they are selective and also differential. Okay? They are selective and differential. Hindi po sila enriched culture media. And mas need po kasi ng Francisella tularensis yung enriched culture media kasi nga po it needs cysteine and thiosulfate. All right? So ayan po. Even though the growth may be observed within 48 hours, plates must still be checked daily for up to 14 days before reporting as negative. So minsan po may mga strains nito na sobrang slow grower. So if we suspect that the organism is in fact Francisella tularensis, we have to wait. Hindi pwedeng after 48 hours or 18 to 24 to 48 hours, i-discard na natin yung plate kasi wala tayong nakita na growth. Okay? So, ayan po. If we highly suspect that the organism is Francisella, dapat po, we would keep our plates for up to 14 days. Now, colony morphology is as shown here on the right. They are grayish-white in color. They are smooth and raised. So, ito po, these organisms or these colonies were observed after 72 hours of incubation. Next is Legionella. All right, so this organism was named due to its historical background. So katulad ng Haemophilus aegyptius, may historical background din po ang Legionella pneumophila. All right, so way back in 1976, in a convention of the American Legion, there were 221 persons that fell ill with pneumonia, and 34 out of those 221 died. Okay, so during that time, hindi pa nila alam kung ano yung cause ng illness. They just knew that the patients presented with pneumonia and some of them died. Okay, the causative agent was later identified as Legionella, and the disease was then termed as the Legionnaire's disease. Kasi po nanggaling siya dun sa convention nung organization na yun. Okay, now there are two forms of the disease. So Legionnaire's disease is a febrile illness with pulmonary involvement. Now, if the patient does not present with pneumonia, the disease is simply known as Pontiac fever. So mas severe po yung Legionnaire's disease, and then it has a milder version or yung mas mabait niyang kapatid, which is Pontiac fever. All right, so the incubation period of legionellosis could range from 2 to 10 days, and clinical manifestations are non-productive coughing plus fever until the disease would progress to the development of pulmonary infiltrates and bloody sputum, which could easily be confused with tuberculosis. Okay, so magsisimula siya na non-specific, coughing, fever, that's it. But as the disease progresses, ayan po, pwedeng mag-produce ng bloody sputum yung pasyente. So pagdating po natin sa following weeks, i-discuss din po natin yung disease progression ng tuberculosis. All right. Ayan. So here is a list of its clinical manifestations. Again, a lot of them are quite non-specific. Fever, headache, myalgia. Diba? May mga nabanggit na rin po tayong organisms kanina that produces the same clinical manifestations. All right. Now, for the milder version of legionellosis, which is the Pontiac fever, the incubation period is also at 2 days after initial exposure. So this one is self-limiting and would last for only up to 5 days. And po, so Legionella pneumophila can be found worldwide and from both natural and man-made water sources. Now, there are many factors that has allowed Legionella to survive in such a wide range of water supply. So, anywhere, kahit saang lugar po na nakikita yung may form of water supply, pwede po tayo makapag-isolate doon ng Legionella. So, they can tolerate chlorine concentrations for up to 3 milligrams per liter. Okay? And as we know, chlorine is common in swimming pools. So, pwede tayo makapag-isolate ng Legionella sa mga swimming pools. Legionella can also continuously multiply even at temperatures as high as 43 degrees Celsius. And then they could still survive at even higher temperatures at 60 degrees Celsius. So, kaya nilang mag-survive sa mga hot springs and jacuzzis. So, swimming pools, jacuzzis, out of the question. They can also adhere to pipes, rubber, and plastics. So any industrial water supplies, respiratory therapy machines that is used in hospitals, and even air conditioning systems, kaya rin pong i-colonize ng Legionella. Okay? So ayun po. In summary, these organisms are very tough and difficult to get rid of. So kahit saan po, or if ever may isang outbreak dun sa isang lugar, marami kayong pwedeng tignan na sources. Yung aircon, if there is swimming pool nearby, yun po. All right. Now, since legionellosis is a respiratory disease, the appropriate sources of specimen would be sputum or bronchoalveolar lavage. And for purposes of public health investigations, in cases of an outbreak, environmental samples may also be collected. So, for example, ayun nga, nagkaroon ng outbreak, pwede pong magkuha ng mga swabs from the air conditioning systems dun sa area kung saan nagsimula yung outbreak. If they suspect na swimming pool or jacuzzi yung source of infection, pwede rin po kumuha ng water samples dun, and so on. Now, if there would be delays in the processing for about 2 hours, refrigerating the specimen is fine. But if the delay will take a few days, the samples must be frozen at negative 70 degrees Celsius. So wala naman pong issue for Legionella if they would be kept frozen for a few days kasi nga po very tough or very resistant to a lot of different things ang Legionella species. All right, so under the microscope, they appear pleomorphic and they are also very weakly staining. Although they are classified as gram-negative bacilli and they can be seen either intracellularly as encircled here. So ito po yung immune cell ng pasyente and some of them have clustered on the inside. Although we barely see them kasi nga po they are very weakly staining. And then they could also be observed extracellularly, as you can see right here in these arrows. So it's quite difficult to detect them via microscopy kasi po yun nga, weakly staining sila. All right, so to be able to effectively isolate Legionella from clinical specimens and minimize the growth of contaminants, specimens must first be treated with acid, as explained right here. So ito po, acid treatment. All right, so although they can survive at a very wide range of temperatures, their optimum growth temp is at 35 to 37 degrees Celsius. So for optimal growth po, and for us to be able to harvest as much organisms as possible, ang recommended pa rin po is that we incubate them at their optimal growth temperature, which is at 35 to 37 degrees Celsius. Now, growth may be already visible within 3 to 5 days. Medyo mabilis na yun for Legionella, but it is recommended to keep the plates for up to 7 days. Okay? So if we suspect na Legionella po yung isolate, hindi natin i-discard agad yung plates within 3 to 5 days. For us to report a negative result, we have to wait for approximately 7 days before reporting as negative. Right? Ayan. And then, take note of the preferred culture media, which is the buffered charcoal yeast extract with L-cysteine. Ayan. So, buffered meaning its pH has been adjusted to 6.9. Alright, so again, kapag sinabi po na buffered charcoal yeast extract, dapat po maisip nyo na agad yung Legionella pneumophila. So here on the left is a photo of an inoculated BCYE agar with no additional antimicrobials. Hence, we would be able to observe heavy growth of other contaminants that were not killed during the acid treatment. Ayan, i-compare po natin dito. If the specimen was both acid washed and plated on a selective BCYE, there would be less contaminating organisms that may be seen, and the Legionella colonies would be observed better, as pointed here on these arrows. So ayan po. And colonies would appear very small, grayish white to blue green, and they are also glistening. So kasama sa descriptions nila yung glistening, which has been described as a ground glass appearance. Ground glass in Tagalog, durog na salamin. Okay, so and po. Wait lang, just for emphasis, this one is a contaminant. These large colonies is a contaminant. These ones that look feathery, these are also contaminants. So nasaan po dyan yung Legionella? Yan po, yung mga maliliit na colonies na tuldok-tuldok. Kaya po siya tinawag na ground glass. Ayan, since they're very difficult to observe in culture, there are different methods naman po for identification of Legionella pneumophila. So other identification methods include checking if the organism requires L-cysteine for growth. Of course, the routine procedure is gram staining, and there is also the use of direct fluorescence antibody, or DFA. Now, if we would choose DFA, the bacterial cells would appear as fluorescent green specks under the microscope, as we can see right here in this example. Ayan po. So, ayan, here they have been magnified for 450 times, and then dito po, ito yung 1,000 times magnification. Ayan po, here at 450 magnification, yung mga maliliit na tuldok-tuldok na kulay green po, yun yung Legionella pneumophila. All right, last but definitely not the least bacteria that we will be discussing in this chapter is Bordetella. All right, so there are two species implicated in human infections, Bordetella pertussis and Bordetella parapertussis. But there are other miscellaneous species that have been associated in a number of various infections, such as Bordetella bronchiceptica, which is an opportunistic pathogen, Bordetella holmesii, which are opportunistic pathogens specifically for immunocompromised individuals. And then we also have Bordetella avium and Hinsii, which are generally animal pathogens. So most likely ma-observe po natin itong dalawang to in animal infections, while Bordetella traumatum have been isolated in cases of wound and ear infections. So for this discussion, focus po tayo dito sa pertussis and parapertussis. All right, so on the table below, taken from our reference textbook, here are the differential characteristics between Bordetella pertussis, parapertussis, and bronchiceptica. Again, most likely hindi po ako yung gumawa no quiz nyo or exam questions nyo dito kasi ako na po yung gumawa no voice-guided discussion. So again, kapag may mga nakita po kayong tables sa textbook ninyo, aralin nyo rin po sila. All right, so for the general characteristics of Bordetella, they are obligate aerobes, they are non-carbohydrate fermenters, and they can oxidize amino acids. And aside from being catalase positive, they are relatively inactive in all other biochemical test systems. So kapag ginamitan po natin sila ng API, halos lahat doon sa API na yun, magne-negative po sila. They can't ferment any carbohydrates. Ayun po, ang kaya lang nilang gawin is either mag-positive sa catalase or oxidize a few amino acids. All right, proceeding with the specific species, first let's look at Bordetella pertussis. So it can easily be inhibited by many compounds, hence they need protectants such as charcoal or blood or starch. Okay, now what are the virulence factors of Bordetella pertussis? The most significant virulence factor out there is the pertussis toxin that has the ability to modify host proteins. Okay, and also andito rin po yung filamentous hemagglutinin, which helps them adhere to surfaces of the ciliated respiratory epithelium. And then they also have adenylate cyclase, which inhibits the action of immune effector cells, and tracheal cytotoxin that causes ciliostasis, or the paralysis of the sweeping motion of the ciliated epithelial cells. So if you would remember, kasama din po yan sa mga virulence factors ng isa sa mga Haemophilus species kanina, di ba po? They also have outer membrane proteins. Ayan, the outer membrane proteins of Haemophilus influenzae functions in the same way. But the difference is that the tracheal cytotoxin of Bordetella pertussis can also cause cell death by inhibiting DNA synthesis. So kung yung outer membrane proteins po, pinaparalyze lang nila yung sweeping motion ng ciliated epithelium, yung pertussis toxin naman po or yung other virulence factors ng Bordetella pertussis, aside from just paralyzing the sweeping motion of the respiratory epithelium, they could also cause cell death. All right, so now let's talk about the phases of pertussis, otherwise known as whooping cough. Okay, so Bordetella pertussis is the causative agent of whooping cough, and this disease has three phases. So first is the Catarrhal phase, which presents with very non-specific symptoms, but they may cause respiratory distress and apnea or cessation of breathing for infants. So medyo mas mahirap po for younger children yung pagkakaroon ng whooping cough kasi po, yun nga, mas maliit yung airways nila. Next is the paroxysmal phase, where we get to hear the characteristic whooping sound at the end of each bout of cough. And this is due to the patient rapidly gasping for air. So if ever nasubukan nyo ng inuubo kayo, tapos dire-diretso yung ubo nyo, it gets difficult to breathe, right? So you would inhale air very rapidly, and then magagaling yung whooping sound. So yun po yung parang whooping cough. Okay, so doon nanggaling yung common name niya. And finally, at the convalescent stage, the coughing spells would decrease in frequency until the patient finally recovers after a few weeks. Ayan po. So next one is Bordetella parapertussis. So all we need to know here is that it causes a disease that is somewhat similar to pertussis, but it is milder. So para siyang Legionella, di ba? Meron siya nung mismong legionellosis and meron siya nung milder version, which is the Pontiac fever. So parang ganun din po dito, except magkahiwalay na organisms sila. Pag Bordetella pertussis, it causes whooping cough. Thus, Bordetella parapertussis, it causes somewhat a similar infection, but it is milder. So for Bordetella parapertussis, it does not have the gene to express the pertussis toxin. So yun po yung main difference ng Bordetella pertussis versus parapertussis. Ang Bordetella pertussis, it has the gene to express the pertussis toxin. Bordetella parapertussis doesn't. So ang meron lang siya is yung iba pang mga virulence factors except the pertussis toxin. Okay? So it does not express the complete functional unit. All right, let's now proceed to specimen collection and handling. Sources of specimen are nasopharyngeal swabs using only either calcium alginate or Dacron polyester swabs. All right, so take note though that these samples may only be used in culture. Okay, so studies show that if PCR is to be performed using the same calcium alginate swabs, the organism was inhibited. So please keep that in mind. Kapag nagko-collect po tayo ng sample at ang gagamitin natin is calcium alginate swabs, pwede lang natin itong gamitin for culture. Pero kung kailangan nyo po ng specimen for PCR testing, it is best that you do not use a calcium alginate swab kasi po makaka-inhibit yon in producing any form of results sa PCR. So most likely kahit present yung organism, magne-negative siya kasi na-inhibit ng calcium alginate. Alright, so the photo right here on the right shows us how a nasopharyngeal swab is done. So this is the same technique na ginagamit po for collecting samples for COVID-19 testing. Okay, so all right, let's proceed to microscopic examination. In gram testing, extending the time of contact with safranin for up to 2 minutes or using 0.2% basic fusion may be used as a counterstain to enhance the visibility of the organism. So for us to be able to observe Bordetella species, i-modify natin yung staining procedure. Other methods would be direct fluorescent antibody method, and this may also be used but should not be considered as a replacement for bacterial culture. Okay? So pwede naman tayo mag-DFA pero mas preferred pa rin kung Bordetella species yung gusto nating i-observe, mas preferred pa rin po that we proceed to culture. Now, speaking of culture, what are the different culture media that may be used for the isolation of Bordetella? All right, so there are two culture media well known for isolating Bordetella species, and these are the Bordet-Jean-Cope data infusion agar and the Regan-Low selective agar. All right, so Bordetella pertussis, or Bordetella parapertussis would generally grow faster, with colonies readily observable within 24 hours of incubation. But Bordetella pertussis would take up to 3 to 5 days to grow. Okay, so yung mild, yung mas mabait na version, which is si parapertussis, within 24 hours, nag-grow na. Pero yung more severe version, which is Bordetella pertussis, ayan, it would take up to 5 days before natin i-consider na negative nga talaga yung isolate. Alright, so before reporting as negative, the plates should be kept for as long as 7 days. Alright, and as pictured on the left, the colonies of Bordetella have been characteristically described as mercury droplets, appearing to be small, shiny, and almost silver in color. So ayan, kapag nakita nyo po na it has been described to appear like mercury droplets, dapat po maisip nyo na agad is Bordetella species. All right, so here's an example of their mercury droplet appearance on culture. All right, so here are the references that were used in the making of this discussion. Ayan, thank you so much po for listening. Our chat box is now open for questions. So does anybody have any questions po?